Good afternoon. My name is Rona Weinstein, and I'm a member of the Department Lectureship Committee. This is a new effort to support department-wide intellectual exchange. A warm welcome to all of you to our annual Distinguished Faculty Lecture, The Question of Color, given by Professor Emerita Karen K, Valois, a renowned scholar of vision. Karen is a former chair of psychology, the first female, and thus far the only female chair, who holds appointments in psychology and optometry. Not only respected for her fierce intellect, Karen is also deeply loved by those who've had the pleasure of working closely with her. We are in for a treat today. The Distinguished Faculty Lecture Series was the idea of former chair Irv Hafter, and it began in 1989, 24 years ago, with the late Richard Lazarus as the first faculty so honored. In my view, this annual lecture accomplishes three things. First, it honors retired faculty members who have served the field and our department so well. Second, it provides a special opportunity to learn from reflections about an entire career of research on a particular topic or topics, the highlights, the low moments, and the ways one's thinking might have changed. Finally, it teaches us about the history of research that's unique to Berkeley, now represented in different ways in our department, or even not at all. It's important to remember that we stand on the shoulders of those who walked before us. They hired you, or they hired those who hired you. I thank our committee, Chair Rich Ivory and Administrative Director Francis Katsura for supporting this lecture in such a beautiful setting and with a lovely reception that will follow Karen's talk today. I invite up Professor of the Graduate School, Steve Palmer, who will introduce Karen K. Valois and her work to you. Following her talk, there'll be time for questions and then on to the reception. Steve.
Thank you, Rona. Karen has asked me to say a few words of introduction about her distinguished career as a vision scientist. Perhaps the first thing you ought to know is that Karen actually began her career as a graduate student at Indiana University in the 1970s, not as a vision scientist, but as an auditory scientist. Pardon? 60s. Oh, excuse me. Why the change to vision science, you may ask? Well, it certainly isn't that she was drummed out of the auditory community, but that she was seduced into the vision science community. But perhaps that choice of words may be a bit too apt. The undeniable facts are that she met Russ D. Valois while they were at Indiana, and that they came to Berkeley together after Karen received her PhD in 1973, right? Yeah, okay. The rest, as they say, is history. I don't think it's an exaggeration to say that Russ and Karen became one of the premier research teams of modern vision science. There's Hubel and Wiesel. There's Hurwicz and Jameson. There's Ungerleider and Mishkin. And of course, there's D. Valois and D. Valois. Russ and Karen shared a deep and abiding passion for unraveling the mysteries of how the visual system makes sense of the three most basic dimensions of visual information: color, space, and motion. Both together and separately, they made groundbreaking contributions to each topic, as well as to understanding the complex relations among them. When I think in terms of the sheer impact of Karen's work, and that with Russ, I'm hard-pressed to think of a single more important and influential work than their joint book, Spatial Vision, which was published by Oxford University Press in 1988. It provided a compelling argument for their hypothesis, once hotly disputed, but now clearly settled in their favor, that area V1 of visual cortex performs what they called piecewise spatial frequency analysis of the visual image. As of this afternoon, Google Scholar says it has been cited more than 1,500 times, a number that I suspect is, if anything, an underestimate of its actual impact. Another integrative work that has attained the status of a true classic was their 1975 chapter on the neural coding of color in the Handbook of Perception, where they described a two-stage model of how the visual nervous system processes color. Their views dominated the field until at least 1993, when they published their revised and more sophisticated three-stage model of color vision. In stressing the importance of their justly famous work, I don't mean to imply that all of Karen's contribution to vision science were collaborations with Russ. Far from it. She's published literally dozens of articles and chapters on her own, and dozens more with collaborators, including Gene Switkus, Mike Webster, Roger Tutel, and Tatsudo Takeuchi, as well as many of her students. And she's done this on a wide variety of topics, ranging from highly technical studies of interactions between color and space, and color and motion, to a paper about seeing bicycles at night. But trying to talk about Karen's accomplishments without mentioning her work with Russ would be like trying to write a biography of Kahneman without mentioning Tversky, of Laurel without mentioning Hardy, or, my favorite, of Rogers without mentioning Astaire. They were a team par excellence, and together they reached heights that neither of them would likely have achieved alone. Now, beyond her impressive scientific credentials, Karen's been a tireless and successful administrative contributor to all of her various academic programs, which include the Psychology Department, the School of Optometry, and the program in Vision Science, all at Berkeley. She was chair of the Psychology Department, as Rona mentioned, and a very effective one from 1998 to 2003. At the campus-wide level, she was also on and eventually chair of the all-powerful Budget Committee. No doubt there are many faculty members in this room right now who owe various positive aspects of their lives and careers to Karen's administrative decisions, whether they know it or not. In recognition of her combination of scholarship and service to the campus, Karen was honored with the prestigious Berkeley Citation at her official retirement ceremony. Here is a description of that honor to be awarded to a faculty member who must be an eminent member of the UC Berkeley School, College, or Department, must also have a record of highly distinguished achievement in his or her academic field. The nominee's work must have had a broad impact within and perhaps beyond the university. At the same time, the nominee must have a record of truly notable service, other than teaching and research, to the university as a whole, or to a significant part of it. Academic achievement and university service of the highest order are required. I think this describes Karen's career to a T. Today, Karen is faced with the difficult task of presenting a retrospective overview of her entire scientific and academic career. Doing that is literally impossible, of course, so she selected a focus that is, in my own view, inevitable. For despite her many contributions in so many areas of interest, the topic that has always been closest to Karen was and is color. Indeed, I think it's the reason she wanted me to introduce her, because she knew I would wear some kind of outfit like this. It's therefore fitting that today Karen is going to provide her own unique perspective on her career with a specific focus on the issues that have been her principal academic passion, the question of color. And now, with apologies for being so long-winded, I'm delighted to introduce our speaker, Karen Kennedy de Valois.
Thank you.
Sweetheart.
No, thank you. I don't know. Thank you very much, Steve. I'd like to dismiss the meeting right now. There's no point going further. He said much nicer things than I could or ever would dream of. I appreciate it. And Rona, I appreciate your words and the honor that your committee and the department have given me. 40 years ago, when I was a whole lot younger than I am now, and I was just beginning my career, Steve was right, I had worked in audition as a graduate student. But I have to say it was not just marrying Russ that got me interested in color, though that did help. Color was a topic that had interested me for a very long time. In fact, if you noticed this when you came in, it interested and bothered Plato and all manner of other people. It's been a topic of considerable effort and interest and concern for literally thousands of years. So in a way, that's very good, because when I was beginning and trying to think about what kind of research I'd like to do, I was certainly drawn to color, partly because color has such a long and deep history. Unlike many areas in psychology, there is a very deep and very profound history of research, of serious scientific work in color. Long before there was a psychology department, there was a history in color. So I was thinking about color when I happened to hear a comment that had been made by a very senior, very distinguished colleague who works in vision, and it nearly crushed me. His comment was, what does color do? You know, the only thing color's good for is letting us appreciate sunsets. Now, I appreciate sunsets with the best of them. But it seemed to me highly unlikely that that was what the color vision system did, or that was its reason for being. Now, I'm not a philosopher, and I'm not going to talk about teleology. I will not tell you what the purpose of color is or why we ended up with color. I would like to note, though, that color vision has evolved independently multiple times in multiple parts of the whole animal kingdom. That suggests to me that there must be something very useful about color. The other thing, or another thing that strikes me as being particularly compelling, is the fact that if you look at the early levels of the visual system, the retina, the lateral geniculate nucleus, you will find that something like 80 to 90%, let's say 80% will be conservative, of all the neurons are involved either entirely or in part in encoding color. That is a massive neural investment. It seems to me highly unlikely that that investment would have been made and maintained over these thousands of years were color not of some considerable use. Now, if you talk to people who study color vision and you ask them, why do you have color vision? They will say things like, oh, you need to be able to tell the ripe fruit from the unripe fruit, or find the fruit hanging on the tree, and doubtless color helps in that. But I find it unlikely that that's the whole reason or the whole thing that color does. So I set out to consider the possibility of trying to understand what color does in a variety of other ways. Now, this picture, Matisse's Woman with a Hat, was painted and exhibited first in 1905 in Paris with works by a group of his contemporaries. This exhibit caused just almost an intellectual riot throughout Paris. People were livid. They were furious. These were dreadful, dreadful pictures. One critic of the day wrote, "A pot of paint has been flung in the face of the public." Not so happy. And Matisse's painting, this particular painting, came in for particular criticism. It was singled out as being the worst of the bunch. Now, Gertrude and Leo Stein, who were then living in Paris, thought Matisse was actually worth following. So they bought this painting, and they kept it in their family for some years, then gave it to a member of the Haas family, who gave it to the San Francisco Museum of Modern Art. So if you would like to see the original painting, it's in San Francisco. Now, that doesn't seem particularly shocking to us, but it was not only shocking, but infuriating when it was first painted. So I wondered, if we were to take this painting and remove all the color, is there anything that we get from the color that we don't get without it? So I did that. Here's the painting. And what you can tell immediately is that the painting is still perfectly recognizable. There is no obvious loss of information other than information about color. It is, however, certainly less interesting, less lively painting. So although I think the painting looks much better in color, we're still left with the question of what does color do? Now, before we can go too much further, I want to just give a very quick review of basic mechanisms of color vision. And for many of you, this will be old hat, and it's okay if you go to sleep, I don't mind. First thing you need to realize is that color is in your head, it is not in the world. As Newton said, the rays of light themselves are not strictly colored. Color is something that we impart to them. Color vision is a 3-variable system, and this was shown experimentally in the mid-19th century. If you wish to match any color, you can do so with an appropriate mixture of 3 and only 3 primaries. That's astonishing. That tells you that there is a 3-variable limitation or a 3-channel limitation at some point in the scene, some point in the system. The next thing that I think is particularly interesting and important to realize is that the same photoreceptors are responsible for producing the input to both the color system and the luminance system. Now, I'm going to use this word luminance. You can think, if you wish, intensity, because as we increase the intensity, we're increasing the luminance. The reason I don't want to say intensity, and we'll use the word luminance, is that luminance is a measure of intensity that's weighted by the sensitivity function of the visual system. The eye is not equally sensitive to all wavelengths that can be seen. So in order to equate things for effective intensity, we equate them on luminance, the scale, rather than strictly intensity. So we know that beginning in the retina, the eye segregates or divides the information about the world into information about color and information about luminance. And we have these 2 pathways coming up. One of the questions I want to ask is, how do the 2 pathways differ? I told you that there were photoreceptors that respond to both color changes and luminance changes. I did not tell you that there were 3 types of them. That's where the 3-variable, well, partly where the 3-variable limitation comes from. We call those L, M, and S for long wavelengths, middle wavelengths, and short wavelengths, which is very unimaginative, but much better than the alternate, which is to give them color names, because they aren't truly color receptors, and I'll show you in a minute. The 3 cones differ in spectral sensitivity, and this will give you an idea of what their spectral absorbance functions are. What you will see now, the dashed line there is the spectral absorbance function for the rods, which I have not mentioned. They're the receptors that work at very low light levels. So when you get up in the middle of the night, you don't turn on the lights, there's not a bright moon, you just have a little bit of starlight. It's your rods that are doing everything for you. You have no color vision, and you don't have very good spatial vision either. But during daylight light levels, or at daylight light levels, you have these 3 kinds of cones. The L cones peak right in here around 565 or so nanometers. And that, my friends, is not red. That is a somewhat yellowish green with a neutrally adapted eye. The M cone peaks around maybe 535, a little bit shorter. Also not a pure green, a somewhat yellowish green. And the S cone peaks way down here in the very short wavelengths. The S cone really doesn't absorb very, very much above about 525 or so nanometers. We customarily call the visual or the visible spectrum, say it extends from wavelengths from about 400 to 700 nanometers. You can see this doesn't extend to 700 nanometers, but it should. That is not to say that there's a sharp cutoff, that you can't see anything lower than 400 and higher than 700. You could see way out into the infrared once. But doing so would damage your retina, so you could not do it again. So this is most of the useful visual spectrum. Now take a look at this. This gives you an idea of what the spectral lights look like in that range. This M and this L cone both absorb over the entire visible spectrum. There is no narrowband sensitivity to any color. So how do we get color sensitivity out of that? Well, we do it by subtracting one from the other. We look at the difference between them. And that then gives us a measure of the wavelength distribution of the light. We can also subtract the S output from a combination of the M and L, or vice versa. By doing that, we will have 4 channels, L minus M and M minus L and S minus L plus M and so forth. We have now gone from 3 cones to 4 opponent mechanisms. That solves one of the big problems that worried people for 100 years. Is it 3 or is it 4? How do we do this? Well, it's both. This is not a big problem. Okay, but they didn't know it at the time. Now, the M and L are not just subtracted from each other, they are added together to produce the luminance mechanism. So each of those cones, L, M, and S, is performing multiple tasks. And the L and M in particular are giving us both the luminance information and color information. So the next slide I want to show you is a little hard to see. Whoops, sorry. Okay. This is an old figure. It was published in about 1966, I think, 65 and 66, by Russ, Jerry Jacobs, and Izzy Abramoff, showing the 6 kinds of cells that they saw in the lateral geniculate nucleus of the thalamus. It's the main way station between the retina and the cortex. I'll pull out this one in a minute and show you a better drawing of it. What I want you to see is that these upper 4 excite at some wavelengths and inhibit in the others. The dashed lines across there are a measure of the spontaneous activity of that cell. Many of the cells in the brain are spontaneously active. They can be either excited or inhibited, and either way carries information. So these cells, these 4 cells, are all now color opponent. They excite to some wavelengths and inhibit to other wavelengths. This just takes one of those cells and shows a little bit more clearly. This is a plus L minus M cell. It excites in the long wavelengths where the L absorbs more light than the M cone, and it inhibits in the middle and short wavelengths where the M cone absorbs more light than the L cones. This slide, incidentally, I took from the textbook of biological psychology that was initially published by Mark Rosenzweig and Arnie Lyman in our department. It's continued through several iterations. It's now being published by Mark Breedlove and his colleagues. And this came from, I don't even remember which version of that text. Okay, so with that in mind, I'd like to look at once more, what the world looks like without color and see if we can figure out anything more about what color might do. This is a very nice picture of a bed of tulips. I don't think you have any trouble noticing that there are multiple rows. Down here is a row of what appear to be bicolored tulips. Here's a row of darker tulips, and there is a row of much brighter tulips. You can see every flower. You can see their leaves, their foliage a little bit. You can see that they differ in color. You have the feeling of having complete spatial information about that image, even though there is no color in it. However, if I put the color back in, you'll notice that there's actually a 4th row of tulips up there. And they are substantially different. They stand out quite a lot. They're pink and white, and you could not see them when we had the color taken out. Now, although you have the feeling that you have complete spatial information about the scene, you in fact may miss considerable and important parts of it. You not only miss them, the color draws your attention to it. Now, with this in mind, I want to go back to the black and white. Now, can you see that 4th row back there? Well, I think you actually can't very well. You see the little bit of white here, and you see a little bit of white there, but I don't think you would ever pick out the fact that there are pink flowers in there. So it's clear that although we get a lot of spatial information with black and white or luminance, and we feel perhaps that we have full information, we clearly don't. So one thing color does is give us spatial and object information that we could not otherwise see or would not otherwise have. Now, I told you that there were these 4 opponent channels, the 2 L and M channels and the 2 S opponent channels. I can analyze this and pull out the LM opponent information from it, and I'll show you in the next slide. It looks a little difficult to see. I think if you look carefully, you can still probably identify these as being tulips down here. I don't think you could identify this stuff up here at all. You might decide it was flowers. You certainly can see that they differ somewhat in color, but you can't identify the color. Now let's look at the S-opponent part of it. And that is, if anything, worse. I don't think you could identify that they were tulips or necessarily even flowers. But if we take those last two, the LM opponent and the S opponent signals, and put them together, again, without luminance variation, now you get a lot more. You can now see a good bit about the individual colors, but you still are missing a lot of spatial information. You can't see the individual stems and leaves. And actually, though you might not think so, you're not getting terribly good color information either. The color information is going to be a lot clearer when we add the luminance to it. Look in particular at this row. When I add the luminance back in, you can see it's a very bright, beautiful yellow, which even with both of the color signals was not completely clear before. So color, in this case, gives us a good bit of information that we did not get just from the luminance alone. But luminance also makes the color a lot easier to see and to determine. Now, so at this point, what can we say that color does? It can draw attention to things that would otherwise be missed. It allows the identification of objects that we could not otherwise necessarily identify. Do we have useful vision based on color alone? Well, honestly, not much. We have some, but it's not good. You would not want to rely just on your color system for your spatial information. So we're going to consider first how the luminance system treats spatial information. And I'll do this very quickly. And thank you, Steve, for mentioning the spatial frequency stuff, because now I don't have to. This pattern of stripes, suppose that you take a sinusoidal grading, a sinusoid pattern, and you just kind of smush it in or pull it out from one side. Or imagine the ambulance that you hear coming towards you, and the frequency rises as it gets closer and falls as it gets further away. Well, that's what this is. This is a visual sinusoidal pattern, but it's now frequency modulated. If we start on the left, the frequency is very low, and it's just rising higher and higher and higher. Now, the contrast starts the maximum contrast at the bottom and falls off consistently until we get to the top. What I want you to notice is this. Suppose we look at this level. Now, depending on where you're sitting, this may be a little bit different, but I suspect you can't see much happening in here. But when you get over in here, now you can see that there are stripes. However, out here, you don't see any stripes. Now, if I asked you to just draw a contour around the area in which you can see stripes and separate that from the area in which you couldn't, again, this is going to be different depending on your distance from it, but I expect it would be something like that. Well, if we actually measured your ability to see each of those individual spatial frequencies, we would get exactly that kind of function. So your contrast sensitivity, your ability to see spatial contrast in a simple pattern in luminance, peaks, at this light level, probably about 2 or 3 cycles per degree, falls off by the time you get up to about 60 cycles per degree. It's history. You can't see very high spatial frequencies. But, and this is interesting, it falls when you get to very low frequencies. This, by the way, that fall at low frequency makes it different from any lens. All lenses will just get worse and worse as you get higher and higher. They'll never fall off at low frequencies. So that's what things look like, or sensitivity function looks like, when we have just luminance. Okay, one thing that we learned over the years was that this doesn't represent the activity of a single kind of cell. It's really the envelope of a bunch of more narrowly tuned mechanisms, which we refer to as channels. So you have a lot of more narrowly tuned spatial channels. which go together to make up this overall contrast sensitivity function for luminance. Now, we didn't do all of that. We did our share of it, but lots of other people did it too. The question of interest now is what happens with color? What if we take out all the luminance contrast and we have a sinusoidal grating that varies in color, but not in luminance? Now things begin to get quite different, because where luminance shows this extension out to about 60 cycles per degree and fall off at low frequencies, color falls off much faster at high frequencies and does not fall off at all at low frequencies. What does this mean? Well, the lack of sensitivity at high spatial frequencies means that we cannot discern the fine details in a pattern if they're based strictly on color variation. The increasing sensitivity at low spatial frequencies implies that color could give us more and better information in this region. So I just did a little experiment. Have any trouble reading that? I hope so. I didn't actually calibrate this. I just did a quick match by eye, so it's not well done. But I've tried to get this as close as I could to an isoluminant pattern so that the red and the green would be approximately equal in luminance. And you see, I think, that it's extraordinarily difficult to read. It's hard to get out fine detail when you have only color there. So, although the color system has some spatial abilities, you don't want to have to rely on it. It does have multiple spatial channels, though they're somewhat broader than those we see for luminance. And a lot of this was work that Gene Switkus and I did together more years ago than either of us cares to remember, and Mike Webster and Arthur Bradley and various other people. We looked at those spatial channels using frequency discrimination and frequency-specific adaptation and frequency-selective masking, and I won't bore you by going into all that. What I want to do is lead to the next point, which is we did the next thing, which is look at color and luminance together. What happens when you start putting them together? Because they're together, of course, in the real world. So we first began just using a simple masking experiment. Now, the psychologists among you will remember Weber fractions, delta I over I, and that sort of stuff. Well, this is a little bit like that, except what we're doing is putting up a pattern of stripes, a mask pattern with some contrast, and then we add to it a little bit more on top of that. And we see how much more you have to have in order to be able to see it. What we're increasing now is the contrast. I don't mean the grading is getting higher. We're making the contrast a little bit higher. And we're seeing how much higher we have to make it for you to be able to see it. And we compare these thresholds to the thresholds that we would get when there's no mask present. So the first thing that we did was say, suppose we want to detect a colored pattern, let's say a red-green grading, in the presence of a black and white or yellow and black mask. So we have a luminance mask. We can try to put a color grading on top of it and see whether we can detect it. Two interesting things. One was that when the mask contrast is very low, it makes it even easier to see the red-green test. Whoa, we're getting facilitation now. That's cool. Once it gets above its threshold, the mask, and gets pretty much above its threshold, it begins to make it a little bit harder to see, but it's not a very strong effect. So luminance can make it more difficult to see color, but it's not a very profound thing. It doesn't get much more difficult. But then we turned around and did the opposite, and my goodness, what a change we found. If we're now trying to detect a luminance varying grading in the presence of a color mask, if the color mask is below threshold, so you really can't see it, we're just going to see whether it adds to the luminance grading. There's no effect. Well, big surprise, who would expect anything like that? But once the mask grading, once that color pattern becomes detectable, it instantly starts to make it harder to see the luminance pattern. And as the color mask gets higher in contrast, the more it just wipes out the luminance test. So we have extremely asymmetric and profound masking. So there's an asymmetric interaction in which color masks luminance much more than luminance masks color. And Jean and I both felt really cool, I think, when we first saw this. And then we had to try to explain it, and then, it wasn't so cool then, but, Okay, so let's go back to the question of what does color do? Well, it does let us appreciate sunsets. It provides some spatial analysis of its own, but it's not very good. It's significantly inferior to what you get with luminance. It nonetheless can enrich and complement a pattern that has luminous differences present. And it makes analysis based solely on luminance contrast more difficult, as we saw here. Now, I didn't design this system, so don't ask me why. Well, at this point, we decided to look at a different thing. Let's look at the effect of color on something different. And we got interested in motion. Variations now not in space, but in time. And if you have variations in time, then you can put time and space together, and you have motion. So we're really interested. We're interested partly because there's some very profound differences between our ability to see temporal change in luminance and our ability to see it in color. If you just take a light and turn it on and off really fast, well, you wouldn't do it by turning the light on and off, but you could. And you could do it many, many, many times a second, and we say, how rapidly can we turn that on and off? And you can see that it's still being turned on and off. We can go up pretty easily to about 60 hertz. 60 times a second, we can turn that thing on and off, and you can still see it. Under really optimal conditions, you can go up higher than that. But you can see pretty fast flickering things when you have a luminance change. But with color, oh no, it's much, much worse. If we substitute, don't turn something on and off, but we have a red pattern here, and a green pattern here, or just a red screen here and a green screen here, and we just alternate them like that. By the time we get up to about maybe 10, 12 times per second, you no longer see it. You just see now a solid yellow pattern. And if we've matched those red and green for luminance very well, you will just see the pattern, the screen being a steady yellow. If we go to a higher frequency, or and the red and green aren't perfectly matched, you'll still see yellow, but it'll go bright, dark, bright, dark, bright, dark. So we knew to start with that there's a huge difference between your ability to detect temporal changes in color versus luminance. We began to look not just at your ability to detect flicker, but at something called the temporal impulse response function. Suppose that I were just to flash a sudden light, brighter light, and I could measure what was happening in the whole system in response to this instantaneously on and instantaneously off bright light. Okay, the response would be very fast. It would rise rapidly to a peak excitation, and then it falls very rapidly, and it goes down into an undershoot, a negative phase, and finally it comes back up to its baseline. If we do the same thing with color, now we don't have a black screen and we're not flashing it white. Let's say we have a white screen and we're flashing it red, so we've matched the luminance. We get something entirely different. First, the latency is longer. It doesn't start until later. It reaches its peak significantly later. By the time the color response has reached its peak, the luminance response has already fallen back down to baseline. And then the color just slowly dies out over time. There is no negative phase with the color. So we have very different functions here. You can't follow rapid change in color. You don't have this fast biphasic response in color. So this made us think about what interesting things that might possibly lead us to. And we thought first, keep forgetting those, of this. Now, in each of those two patches, you see a section of a sinusoidal luminance grading. Do you see it moving upward when it comes? What you're actually seeing is two gratings are being presented. First, it's here, and then it's here. Notice this black bar along the bottom here. In this second frame, it's moved up a little bit. That's why you see the motion upward. So we just have 2 frames of each one, first position, second position, or first phase, second phase. And you see very nice motion upwards. This, by the way, is exactly the kind of motion that you see in all television and movie screens and all. It's all just substituting still frames. Okay, now in the next slide, I'm going to make a change. Right over here, I'm going to stick in a blank frame in the middle. You will not see the screen go blank. It'll be much faster than you can see. But there'll be frame 1 and then a blank frame of the same average luminance, and then this side will stay the same. Let's see what happens. Do you see a difference? Okay, this one still appears to move up, but this one should now appear to move down. Why is it? We have not changed the phases of the 2 frames of pattern. Look here, the dark bar is still here, and the second frame, it's moving up, in the third frame. But we put this blank frame in between. What's going on? Well, I'll explain it to you in just a minute, but before I do, I'd like to show you what happens if we take away the luminance contrast and make these colored patterns. And the colored patterns are going to be just like this on the left side and this on the right side. Remember, here's where we get upward motion on the left here and apparent downward motion here. Okay, now we have exactly the same. The blank frame in here. And you see this one, I hope, going up. How does this one appear? Okay. Okay, we're getting a mixed response, which is that's exactly what we should get, because one of the problems with trying to do this is I can't calibrate it, and particularly I can't calibrate it for your independent eyes. But if you were seeing that in isoluminance, you would see the one on the right going up just like the one on the left does. Yeah, you're perhaps aware there's a little bit of a delay there, and there is a little bit of delay there. Now, what causes that? Why do we get a reversal in direction with the luminance, but not in the case of color? In this case, ideally, what you would want is for this to go up both ways, because that's the way the pattern's moving. Well, let's go back and see whether we can figure it out. Remember that the luminance has that biphasic response. It gives a quick excitatory response and then a negative response. The color is monophasic, it's slow, and it doesn't have that negative thing. So what happens with that negative response? Well, when it goes into that negative response, you are in effect getting a negative afterimage. So now you had a black bar here, but during the blank frame, the black bar is up here. Now, instead of comparing this to this, which would be upward motion, you're comparing this to this, which is downward motion. And so because you have a fast biphasic response for luminance, you're making an error in your perception of the direction of motion. Because you do not have a biphasic response in color, you continue to perceive it correctly, even though we put in the blank frame. Now, this got us thinking about image compression and what we know about image compression. Television engineers have known for a long time and have used the fact that that the color response is very slow, that you can't see things happen very fast in color. And so they sample the color information much more sparsely than they sample the luminance information. So on every frame, they send out luminance information. They don't send out the color information in every frame because you can't tell whether it's there or not. And this is one of the reasons that we have to. This luminance is so fast. But now, you know, I've kind of misled you a little bit, because you don't get that fast biphasic response for all luminance patterns. There are 2 conditions under which you don't. One is if you look at high spatial frequencies, there the luminance pattern shows a slow monophasic response, just like color. And if you go to very low light levels, it does the same thing. It has a slow monophasic response. Tatsuto Takeuchi and I were interested in this problem, and we actually made some quantitative predictions about how this effect should change as we go to lower and lower light levels. But that's got to be one of the most boring experiments in the entire world. You have to dark adapt forever, and it's just, so I'm not going to bore you with that. It worked, I'll say that. But this we found particularly interesting, because this is something that could conceivably work under high luminance levels. So this is exactly the same figure I showed you a while ago. I just changed the labels. They're both luminance, but this is low spatial frequencies, and this is high spatial frequencies. So Tatsuto and I began to wonder whether we could perhaps come up with a kind of image compression based not on the difference between color and luminance, but on the difference between low and high spatial frequencies. So we began with a movie made up of 100 frames. This is a movie of a flying eagle. And I've set it so it just goes round and round, it loops. Okay, so get a good idea of what that looks like. It's pretty well focused, but it's hard to keep perfectly focused on something moving that rapidly. Okay, now we're going to take some proportion of those frames. And we tried things from 50 to 90 of the 100 frames and low-pass filtered them. We took it apart, low-pass filtered some of the frames, then we put them all back together, made the movie again. And we showed the movie now in which some frames were unfiltered, the original frames, and others were low-pass filtered. I'll show you one example of it. This is the same as this frame, but this has been put through a very stark low-pass filter. You can see that not only have we taken out all the detail, I don't think if you didn't know what this was that you could necessarily identify that as a bird. Now, 70 of the 100 frames are low-pass filtered, exactly like this. This is the original movie. There is no filtering there. So let's see what it looks like. I frankly can't reliably tell a difference. The only difference that I can see is maybe a little bit less blackness in the wings. But, pardon? Oh, I think that's actually a problem with the movie because it's normally not. Sorry, I don't know what happened between. But in fact, you can easily throw out all the high frequency information from 70% of the frames. You can actually go up to about 80-85% without much noticeable difference. So we were curious then about how significantly we could blur them. How low down can we take our low-pass filter? How much of that information can we throw out? And I'll tell you before I show you, all of these are going to be flickering. And it has nothing to do with the fact that I filtered it. It has to do with the fact that somebody put this together to run at 15 hertz. So sorry about that. But this is the original here. And as we go down, the blur gets increasingly strong. That means the corner frequency of the low-pass filter gets lower and lower. So take a look. And despite the fact that they're all flickering, what I think you'll see is that you can still see all the fine detail, including the individual spots in this running cheetah. And these 50 of the 100 frames are blurred. So this has nothing to do with color, except that we're just copying what we knew had been done with color and playing a game with luminance. But I've got to say, it was really a kick. It was just fun to do this. You know? It actually worked. So now let's go back to color. We've been thinking about color and motion, and in the example of the moving gratings, color gave the right answer and luminance did not give the right answer. So let's look at the other examples of the influence of color on motion. We did some experiments where we had motion with only color information. We looked at motion with filtered luminance patterns, and we looked at motion with both color and different kinds of luminance information present. This is just an example of one of our movies. This is the original. This is the luminance salon. This is the LM opponent signal. This is the S opponent signal. So we could analyze each one into the outputs of these various channels. Now, what I'm going to show you is the task. We took a bunch of movies which had a clear directional component to them. So, you know, you can tell whether that's being played forwards or backwards. Horses don't generally run backwards like that. And billiard balls rarely go in and form a nice little group by themselves. So all we asked our subjects was to look at the movie, tell us whether it's being played forwards or backwards. Very easy task. Now, I'll tell you in advance, this next couple slides, you're not going to see it very well because there really are some luminance artifacts in the projection here. But this is an example. This began life as an isoluminant pattern. So we just took out all the luminance and we asked the subject whether the horse is running forward or backwards. Have a... Yep. Now we'll look at the next one. And that, of course, is the opposite. The horse is going backwards. Now, the next one, you'll see only the luminance information, but we've done a high-pass filter. So we've thrown out the temporal low frequencies and kept the temporal high frequencies. I particularly like this one. It always reminds me of an insect who gets turned on its back and all his little legs are moving as rapidly as he can make them. So we would ask subjects, is it going forwards or backwards? And they'd say, yeah, I don't know. But then we put those two together, that temporally high-pass filtered luminance and the isoluminance information, and we asked the subject again, is it going forwards or backwards? And I don't know why this is dying on me. Oh. Well, we'll hope it comes back. It's not going anywhere. It doesn't matter. I can tell you all the rest.
Your antivirus?
It's probably, yeah. But let's see. Okay, yeah. Yeah. Okay. Yeah, it's still not going, but it's all right. The fact is that when we did this, we gave our subjects a variable amount of time to look at the film. And we said, okay, you can go anywhere from half a second up to about 4 seconds, we gave them. And when we restricted them to a half second, well, it's hard to tell anything about motion in a half second, they performed a chance. When we gave them the isoluminant pattern, just color, they got up to about, 60% correct by the time we got them out to 4 seconds. We gave them this high-frequency, high-pass filtered luminance pattern. They did exactly the same. They got up to about 60% correct after 4 seconds. But when we put the two together, they showed considerably better performance. By 4 seconds, they were up at 85 or 90% correct. So we showed a considerable improvement in one's ability. Go ahead. To make that judgment. Okay, so in this case, color can replace some luminance information, and thank you, you're magic, and aid in motion judgments. Thank you. I have to tell you, I started doing this with a new computer and knew everything. And the new software would not accept my motion files. So I finally had to go out and pull out this old computer, which is running 2003 office. So it's, it's on its last legs. All right, so I showed you then what happened when we added the high temporal frequency luminance and the low frequency color. This is just a little demonstration. In this picture on the left, we've treated each individual pixel separately, and 80% of those we high pass temporally filtered. 20% of those we low pass filtered. This is just the isoluminant color component. They're low pass temporally filtered. Your eye would do it if we didn't. You can't see that. And this is where we have added this and this. Now I'm going to set them into motion. You can see this is not a very appealing pattern. You can see it move, but it doesn't look very good. It's a flyover of San Francisco. This I don't think you could recognize at all. And you'll see what a difference it makes to add the two. It is, I think, substantially improved by adding the color to that not terribly good motion signal. So what does color do here? In the case we described, it can replace some luminous information. It can make motion judgments better. And it's potentially useful, therefore, in image compression, which is a very important thing in today's world. Okay, one other thing. Okay. We want to know whether color could affect motion perception in other ways. So we took a pattern. Here we have a horizontal grading, and we started that grading moving upward. Now, this shows what happened to the color of it across time. First few frames, it would be pink, and then it would be green, and then it would be pink, and then it would be green. The grading kept moving up consistently the whole time. So this just shows you we have pink and green and pink and green. But as this goes, as the color changes, the pattern is still moving upwards. And so what we're asking our subjects is, how much contrast do you have to have there in order to tell what direction it's moving? Now, this is an interesting test, I think, because this is a situation that does not normally happen in nature. All of the other things that we've seen, one way or another, they might be natural, but this is not. This is clearly an artificial stimulus. And what we found, these are the colors that we used. We would use, this could be the reference, or this, or this, or this. And if, let's say that this was the reference, this color is right here. This is the red end, the L end of the LM axis. So it could go up, and this just shows the contrast in the S cone, this in the L cone, and this in the M cone. Now, let me show you how it looked. If we started with this as our reference pattern and we alternated this with itself, then of course it had no effect. But if we alternated it with this color, which is just a slight bit bluer, had a little bit of effect. You got some masking there. And the further away we got, until we got to 180 degrees, which is the opposite end of that LM axis, and there it would increase the threshold by about a factor of 4. And as we go back down to the 0, falls again. So it follows very nicely along with the contrast in that LM axis, how different the two ends are. But, and this is very interesting, I think, when we used 90 degrees, now this is the plus and minus S axis. So this is one end of the axis, and this is the other end. When we alternated between this color and that color, so we had no contrast in the LM system, we had no masking effect at all. There was a big color difference, going from a purplish here to a chartreuse here, and it doesn't mask at all. The amount of masking is predicted exactly and completely by the amount of contrast in the LM system. So we're now seeing a very powerful masking effect that is color selective. It doesn't happen universally or uniformly for all colors. This, I think, is potentially a very useful thing to have, because in nature, when color changes fast like that, it is generally because you're crossing an object border. You're crossing something in the object is changing. It's not because of the illuminant. So one other thing, and this is the last thing I'll tell you about. I have talked all this time about a luminance channel and a color channel or a color system. But I had wondered for a long, long time too, whether there might be something else. Are there luminance mechanisms, things that look at differences in bright and dark, but that are color selective? Now, it's a little bit heretical, I suppose, to talk about something. Just the very term luminance implies indifference to color. But Joe Hardy and I were interested in that and wondered whether there might be some system that directly combines the two. So we used the Blakemore-Sutton effect. I do not want you to adapt here. This takes too long, and these are square waves anyway. But suppose you look at these two gratings. They are identical. They have the same frequency. This is higher and this is lower. If I were to have you adapt by moving up and down along this line for a while, and then look down here, this grating would now appear lower in frequency, and this would appear higher in frequency. Each one gets shoved in apparent frequency away from the frequency of the adaptation pattern. So that was the basic thing we did. We alternated, we alternated between a high frequency red luminance varying adaptation pattern with a low frequency green luminance varying pattern in the same place. So, well, actually I can show you in just a minute, but when you view a test grating of an intermediate frequency, if the mechanism responsible for the adaptation is blind to hue, the two effects should cancel. And we chose frequencies in contrast such that they should cancel perfectly. And we showed that they did. So during the adaptation pattern, the subject would see something like this, alternating. High frequency when it was red, low frequency when it was green on the left side, low frequency on the right side, and high frequency on the right side when it was green, okay? So you just alternate between these two. And then during test trials, they would get either a green screen or a red screen. In either one, the pattern on the left was always an intermediate frequency, and the pattern on the right was variable in frequency. Simple question, which one has a higher frequency? And from that, we can calculate how much of an adaptation effect there is. The actual frequencies we used, incidentally, were 1 and 4 cycles per degree with a 2 cycle per degree test. If a hue selective mechanism is involved, there should be opposing after effects depending on the hue of the test grating. We found that there was, in fact, an after effect that was selective for the hue. It accounts for about 1/3 of the total effect. When black and white patterns are used, it does not transfer interocularly, but it does not also transfer across orientation. The fact that it does not transfer across orientation implies a cortical origin. So here's just a little bit of data. In this case, when the screen was red, the subject saw a high frequency pattern on the left and a low frequency on the right. When the screen was green, the pattern on the left was low frequency and the pattern on the right was high frequency. This would cancel out any effect that was hue indifferent and reveal only the hue selective effects. What you see is that when the screen was red, just right here, the intermediate frequency appeared shifted to lower frequencies, which is what we would expect. When the screen was green, it appeared shifted to higher frequencies. So we're getting two opposing frequencies from adaptation in the same retinal region, two frequencies, and this effect does not transfer across orientations, so it must be cortical in origin. If we had them adapt to only one color, let's say we showed them an adaptation pattern when the screen was red, when the screen was green, there was no adaptation pattern. Now, when the test pattern is on a red screen, it's pushed further down the frequency. When it's on a green screen, it pushes in the same direction, but not as far. Those are exactly what we would expect. And this is just showing you that there is no adaptation effect at all when we change the orientation. So, and that's if we look at the amount of orientation transfer, the after effects, the hue selective thing accounts for about 1/3, and the hue non-selective about 2/3 of the total effect. So we have demonstrated the existence of a mechanism that analyzes spatial luminance varying patterns in a hue-dependent manner. I do not know of any earlier or any other demonstration of this. The selectivity for orientation implies that it's cortical in origin. This is counter to an expectation of total object constancy. So if you think what the visual system is doing is working entirely for object constancy, it shouldn't be doing this. However, I think it makes sense that it should do that, because you don't want that kind of constancy, I think. So is color only for sunsets? Well, color makes an image much more interesting. It can be used to make parts of an image visible and take your attention. It can improve the appearance of a moving image. It can, in fact, improve your judgments of a moving image. It can aid in image compression. It can prevent errors in motion analysis, and it can segregate spatial analysis by hue. So color is more than for sunsets, my friends, but enjoy the sunsets. Thank you.
Thank you, Karen, for a fascinating talk. We have some time for questions from the audience, and I'll hand you the mic. Questions, responses. Anne.
I'll just speak loudly. Karen, thank you for a great talk. And I wonder,
Thank you.
with all the findings you presented, and there were a gazillion more you didn't have time to, I wonder what were some of the surprising things that came out that you didn't predict? You told us a very linear story here, but
sure.
it seemed that at a couple of times you were surprised by some of these.
Oh, lots of times I'm surprised by things. The first stuff that Jean and I did about 30, 40 years ago, 35, way too many years ago to count. When we first found those strongly asymmetric interactions between color and luminance, I wasn't completely surprised, but kind of. You think of luminance as being clearly the more powerful signal in many ways, and suddenly the color system is just saying, go to bed, I don't want to see any more of you. So it was very interesting, I think, to find that. It was also very interesting to find that the simplistic notions that I initially had about how luminance might make it easier to see color were not right. I mean, there's just some of our early ideas were just completely wrong. I certainly did not expect any of the motion stuff. There had been claims for, I suppose, 10, 20 years that you could not see motion at isoluminance. And it's true that if you have good isoluminance and you're using a tritan pattern that only stimulates the S-opponent system, it looks really weird. I'd like to say you can still see motion. I think it is now generally accepted. I think most people realize that you can see motion, but clearly it is not just a color copy of the luminance signal. I was certainly surprised to see that color could have such a profound effect, even on luminance motion, which was something that I would have thought would be immune to it, but absolutely wasn't. And the most fun of all, frankly, was the image compression stuff, because that was just a kick. It was like, woohoo, we found something new. Yes. And it's also practical. And as I think Rona said, or maybe it was Steve, I actually did one applied experiment in my entire career, and that was the bicycle thing. So I was very happy to have the image compression come out with something usable and surprising. Celine?
So as you studied and you used subjects, and how much did you have to allow for people really do see differently? Was it huge? Was it huge
Well,
in some areas?
No, when we bring in a subject, okay, the question is how much did we have to allow for individual differences among subjects? We don't do the experiments the way a lot of labs do. When we bring in a potential subject, the first thing we do is take him down to the clinic, get his vision checked out, make sure there's nothing terrible going on there. Then we check his color vision, make sure he has normal color vision, and then we do a whole series of calibration things with the individual subject. So when I would make an isoluminant pattern, it would be made exactly for your eye, not for mine or for anyone else's. And then we would use the same subjects every day for months and months and sometimes years. I had a few subjects who worked for me every semester for years. They were undergraduates. They would come in for an hour a day and work as subjects. So you're right, there are differences, and you really better chase those out pretty early before you get very far along. Otherwise, you're wasting time and money. So we did that early. Mm-hmm. Jim?
I suppose you didn't need the variable of colorblindness in your experiment. Did you run any of these on people who could only see blue, for example?
Well, I don't know that there are any people who can only see blue, but we did once. We used a colorblind subject, and I'll tell you what it was. There's a real problem in working with patterns at isoluminance or nominal isoluminance, because the optical system of the eye, though it's very elegant in many ways, in other ways it's inferior even to a cheap camera. There is no color correction in the optics, in the eye's optics. And as a result, you get aberrations and you get unintended luminance traces that you didn't want. So we did a lot of song and dance to try to prevent those as much as we could. But once we actually brought in a deuteranope. I think it was a deuteranope. A deuteranope is a dichromat. A person's missing one cone type, and his luminance function, luminance sensitivity function, is very close to that of a normal observer. So we took the settings that we had, the luminance settings that we had made for a normal observer, forced them on the deuteranope, just said, you know, this, I mean, he didn't know what we were doing, but did not allow him to make independent settings. And then we looked to see whether there would be any sign of luminance intrusion, because he would have the same chromatic aberration that anybody else would have, but he would not have the chromatic sensitivity of someone else. So if there had been significant luminance intrusion, he would have seen it, but he didn't. And so we breathed a great sigh of relief. But other than that, no, I kind of tell those people thanks, but no thanks when they come. Yeah. Bob?
So one of the things that's really remarkable about this talk is we're seeing a field where so much progress has been made and so much of it you were a part of. And so much is known. So I was thinking a student comes in today and says, I want to work on color with you. And you either say, oh no, we're done.
No, I say I retired.
But you didn't say that. I was
Yeah.
wondering, what are the big questions that are, you know, if you had 50 more years.
Sure. Well, that's a good question. There are a lot of them, and it brings me to one point that I meant to say earlier and didn't think about, and that is that I firmly believe that color vision is likely to be the first complex behavior that will be completely understood in terms of the underlying physiology. And one of the things that has driven me all along is that I'd like to be part of that, you know, when it gets there. But are there still questions? Of course. I mean, some of them are the kind of nitty-bitty questions that only somebody like me could love. But some of them are much bigger and more important. I heard a discussion on I think it was Science Friday on NPR recently, asking whether people who have 4 cone types are tetrachromatic. Do they require four primaries to match any color? I didn't mention this, but the L and the M cones both come in multiple varieties. There are slightly different photopigments with slightly different spectral sensitivity functions. And women in particular can have 2 of the same two L-cone types, let's say, with slightly different photopigments. And this raises a question of whether a three-variable system is adequate to describe theirs. Now, I thought that question had been put to bed a few years ago because people did some very serious studies and found that even with those people, the three-variable limitation still seems to be there. What that would suggest is that the narrow part of the pathway is not the cones themselves, but the pathways into which they feed. But at least on Science Friday, they're suggesting that this is still possibly a live question. So that's something I would be very interested in seeing the answer to. My interest at this point, if I were going further, I think I would be really interested in looking more at this last thing I talked about. What would appear to be a luminance pathway, something that's responding predominantly to intensity variations, but is hue selective, because that doesn't fit. I mean, it's just not in our sort of standard model. I don't think it's that hard to see how it could happen, how you could make it. But I frankly was very surprised to see those results and to see that it seemed to be pretty powerful. So I would say, sure, go look at something like that. That's really interesting, because that really could revise a lot of the way we think, I think. But mostly, yeah. I mean, you get into it, you're going to have your own questions, of course. But yeah, those are some directions I would want to go. But I don't want to at this point.
Thank you, Bob, for that question in terms of looking to the future. And thank you, Karen, for a very lively, creative, and inspiring talk. I have a question maybe others might have and can ask as we move to the reception. Now it's tied neatly together, as Anne said, as you look at the whole program, and one wonders what you are up against. In other words, the prevailing theories that you differed from, how difficult it was to publish when the field changed, because it's very much of a dynamic experience.
I have to tell you, many years ago, I found a notebook that my grandmother had had. She had taken a psychology course when she was an undergraduate in 1901. And so, of course, I had to read it. And I was astonished to find that she had 4 pages of notes on color vision. I was particularly astonished to find that all 4 pages were devoted to the model of Ewald Hering. Now, this made me very happy because I'm a great fan of Hering. But for 100 years, there was a huge debate, a real clash between the followers of Helmholtz, who talked about a 3-variable system, and the followers of Hering, who talked about a 4-variable opponent system. And that problem was put to rest shortly before I got into the field, and it was put to rest largely by the physiology. Hurwitz and Jameson did wonderful psychophysics on it, but then Russ's physiology in monkeys, I think, really put the done onto that question. So in a way, it was a good time to get into color. It was a good time for another reason that is, I think, often overlooked. There were technological breakthroughs that came along early in my career. Traditionally, color was studied with optical systems. And with an optical system, you get wonderful control of wavelength and intensity and all that stuff, but it's awfully hard to make a complex spatial pattern. So lots and lots was done on color mixture, color discriminations, color sensitivity, those kinds of things. But the field of spatial vision in color was wide open. And for the first time, with color televisions and computers, for the first time it was possible to do those experiments. So in a sense, we had a really good opening, a really good time. Was there a lot of kickback? No, I don't think a lot. There were certainly disputes and disagreements over the years. Some of you may have read that David Hubel died yesterday or 2 days ago or something. At one point, Hubel and Marge Livingston, who was one of his colleagues, suddenly discovered psychophysics. And they published a really long paper rediscovering all manner of things. So it's like, God, I wish you'd read the literature. But that was part of what became a big question of discussion of these parallel pathways and how the world of vision was divided among them. Russ and I had one strong position on that and still think we're right, so. Which is to say that things are multiplexed, that you don't have just color and just luminance. They go together.
On that note, let's give a hand of applause.
Thank you very much.